Marthe Gautier's story. The city of Paris in the 1950s was a place of electric contradictions. On one hand, the scars of war were still etched into its boulevards and the collective memory of its people. On the other, it was a crucible of intellectual and artistic rebirth. In the cafes of Saint-Germain-des-Prés, existentialists debated the meaning of freedom, while in the city's laboratories and hospitals, a new generation of scientists, fueled by post-war optimism, was poised to unravel the very blueprint of human life. This was a world on the cusp of the atomic age, the space race, and a revolution in medicine. It was in this vibrant, challenging landscape that a young, determined physician named Marthe Gautier began a journey that would lead her to the heart of a profound genetic mystery. Born in 1925, Gautier was part of a generation of French women who were cautiously stepping into professional fields long dominated by men. She was brilliant, focused, and driven by a deep-seated empathy. Her chosen field was medicine, and she quickly found her calling in pediatrics, specifically pediatric cardiology. At the renowned Trousseau Hospital in Paris, she dedicated herself to treating children with congenital heart conditions. Day after day, she confronted the fragility of young lives, working to mend tiny, malformed hearts. It was here, in the course of her clinical work, that she repeatedly encountered a particular group of patients. These children, often born with the very heart defects she specialized in, shared a distinct set of physical characteristics and developmental delays. At the time, their condition was known by the deeply unfortunate and scientifically baseless term Mongolism. Today, we know this condition as Down syndrome, but in the 1950s, it was a fortress of scientific ignorance, surrounded by a moat of social stigma. The prevailing theories about its cause were a bewildering mix of speculation and prejudice. Some scientists clung to the idea of racial degeneracy, a throwback to archaic and offensive anthropological concepts. Others pointed to factors like the mother's age, suggesting some kind of uterine exhaustion in older women. It was a condition defined not by what was known, but by what was unknown. For families, a diagnosis was devastating, often accompanied by shame and the recommendation to institutionalize the child. For scientists, it was a frustrating puzzle. They could describe the symptoms, but the root cause, the biological why, remained completely elusive. This lack of a definitive explanation was a critical barrier, preventing any real progress in understanding, treatment, or even compassionate social acceptance. This is the world Marthe Gautier stepped into. She wasn't just a detached researcher in a lab. She was a doctor who held the hands of these children and spoke with their worried parents. She saw past the clinical label to the individual, and her scientific curiosity was ignited by her clinical compassion. She wanted to know, why did so many of these children have heart problems? What was the underlying biological mechanism that connected all their symptoms? This question led her to the door of Professor Raymond Turpin, a leading figure in French genetics at the nearby St. Louis Hospital. Turpin was a man obsessed with the puzzle of Down syndrome. For years, he had nurtured a bold hypothesis that the condition was caused by a chromosomal abnormality, a mistake in the very genetic script of life. He suspected an excess of genetic material, or what he termed aneuploidy. It was a brilliant idea, but Turpin and his team were stuck. They lacked the one crucial, indispensable tool needed to test it, a reliable method for culturing human cells and viewing their chromosomes. The field of human cytogenetics was in its absolute infancy. In fact, it was only in 1956, just 2 years before Gautier's pivotal work, that scientists Joe Hintio and Albert Levin had definitively established that humans have 46 chromosomes, not 48 as previously believed. Their breakthrough was made possible by new techniques for preparing cells, techniques that were still more art than science, notoriously difficult to replicate. Turpin's lab had tried and failed to master them. They could theorize all day, but they couldn't see the evidence. They needed someone with extraordinary technical skill, patience, and the tenacity to tame the notoriously fickle process of human cell culture. They needed Marthe Gautier. Recognizing her potential, Turpin offered her a research position. The goal was simple to state, but monumental to achieve. Set up a functional cell culture laboratory from scratch and use it to examine the chromosomes of children with Down syndrome. For Gautier, it was an irresistible challenge, a chance to bridge her clinical work with fundamental research and potentially solve one of the greatest medical mysteries of her time. She accepted, unaware that she was about to step into both the scientific limelight and, eventually, a long, undeserved shadow.
Chromosome 21 Discovery. The task facing Marthe Gautier was not merely difficult, it was, by the standards of the day, nearly impossible. Setting up a cell culture lab in 1950s Paris wasn't a matter of ordering a kit and following a manual. It was an act of scientific improvisation, a battle waged on a microscopic scale against contamination, equipment shortages, and the sheer biological stubbornness of human cells. The laboratory space she was given was rudimentary, almost comically so. It was a small, cramped room, a far cry from the sterile, high-tech facilities of today. Lacking institutional funding for her specific project, Gautier became a master of resourcefulness, reportedly using her own modest salary to purchase essential glassware and chemicals. She was not just the lead scientist, she was the architect, technician, and sole operator of her entire experimental setup. Her first and most significant hurdle was mastering the art of tissue culture itself. To test Turpin's hypothesis, she needed living, dividing cells taken from children with Down syndrome. She obtained small tissue samples, fibroblasts, from her patients at the Trousseau Hospital. But keeping these cells alive and growing outside the human body was a black art. The growth medium, the nutrient broth the cells needed to survive, had to be mixed by hand. It was a delicate cocktail, often containing bizarre-sounding ingredients like rooster plasma and embryonic extracts, each batch slightly different from the last. The greatest enemy was contamination. A single stray bacterium or fungal spore, invisible to the naked eye, could land in a petri dish and ruin weeks of painstaking work overnight. Without the modern laminar flow hoods that provide a continuous curtain of sterile air, Gautier had to rely on meticulous technique, alcohol lamps, and a healthy dose of luck. Recognizing the need for specialized training, she secured a year-long fellowship to study in the United States, working with leading cell culture experts at Harvard. There, she absorbed the latest, most advanced techniques, honing the skills that would be indispensable for her mission back in Paris. When she returned, she brought not just knowledge, but a renewed determination. She began her work in earnest, a solitary figure spending countless hours in her small lab. The process was a testament to her patience. First, she would carefully prepare the tissue samples. Then came the long wait as she nurtured the cells, coaxing them to divide and multiply in her handmade culture flasks. But getting the cells to grow was only the beginning. The real challenge was seeing the chromosomes locked inside their nuclei. This is where Gautier's most critical innovation came into play. The 46 human chromosomes are normally a tangled, spaghetti-like mass within the cell's nucleus. To count them, you need to catch a cell at the precise moment of division, when the chromosomes condense into distinct, X-shaped structures. Then you need to spread them out. Gautier perfected a technique known as hypotonic shock. Just before fixing the cells to a microscope slide, she would expose them to a solution with a low salt concentration. By the principle of osmosis, water would rush into the cells, causing them to swell up like tiny balloons. This sudden expansion had a miraculous effect. It forced the tangled chromosomes apart, scattering them across the slide like stars in a tiny galaxy. This single, elegant step made the previously impossible task of counting them feasible. With her protocol finally established, culture, shock, fixation, and staining, the moment of truth arrived in 1958. Slide after slide, she prepared samples from her patients. She would sit for hours, her eye pressed to the brass eyepiece of her microscope, scanning the field of view, painstakingly counting the tiny, stained specks of genetic material. 1, 2, 3, 44, 45, 46. In the control samples from individuals without Down syndrome, the number was always the same, 46. It was the beautiful, consistent number of humanity. Then she switched to the slides prepared from the cells of a child with Down syndrome. She began to count again, her focus absolute. 44, 45, 46, and 47. There it was. A pause. A disbelieving blink. She recentered the slide, adjusted the focus, and counted again. 47. There, amongst the others, was an unmistakable, tiny, extra chromosome. It wasn't a fluke. Science demands repetition. Gautier meticulously prepared another sample from a different patient. She repeated the entire laborious process. The result was the same, 47. And again with a third patient, 47. The evidence was undeniable, consistent, and revolutionary. Turpin's hypothesis was correct. She had found the biological cause of Down syndrome. It was the first time in history that a specific, visible chromosomal abnormality had ever been linked to a human developmental condition. It was a discovery that would rewrite textbooks and transform the field of genetics forever. In the quiet solitude of her lab, Marthe Gautier had unlocked a fundamental secret of human biology, a breakthrough born not of a single flash of genius, but of relentless dedication, technical brilliance, and a profound desire to understand.